Lab 26, Mitosis in Animals. Introduction. Each human starts out as a single fertilized egg. At the moment of fertilization of this new human is only one cell in size and contains genetic information from both its mother and father. This single cell divides into two daughter cells, which then themselves divide. This process continues to form a multitude of cells. As growth continues, they sort themselves out into groups to form the different organs of the new body. Each daughter cell retains some characteristics and loses others. As if by magic, each cell knows its destiny. Some cells group together to form muscle, while others become nerve cells. Scientists are just beginning to unravel the complex genetic and molecular codes that make sure each organ develops just right. The nervous system is the first major part of the human body to develop. The process begins just 2.5 weeks after fertilization. By the 4th week of development, the heart begins to beat and a primitive circulatory system can be seen. Also, at this time, the beginning of the respiratory system, digestive system, urinary system, and reproductive system can be seen. The skeleton appears by the 5th week as special clusters of cells in different parts of the body. By 8 weeks, all major organs are present in some form. It can be clearly recognized as a human. It has a torso with arm and legs, fingers and toes with nails. At this stage, it is about 1.25 inches in size. Over the next 8 months, the cells will continue to divide and the baby will increase in size. The organs develop and mature as the baby prepares for life outside the mother's body. Growth appears to slow down in the last few weeks of pregnancy. It is unclear why this happens. About 5 weeks before delivery, the baby takes control of the pregnancy. The baby has its own program regarding how long the pregnancy should last. It passes this information on to the mother. He calls upon several systems in the mother to help successfully complete the birth process. But the timing of the birth is determined by the baby. After birth, mitosis continues to add to the number of cells in the body, allowing for continued growth until maturity is reached and growth stops. Even in adults, billions of cells still divide. For example, cells of the stomach lining divide every day. Liver cells usually do not divide. But if part of the liver becomes damaged or diseased, numerous cell divisions will yield new cells until the damaged parts are finally replaced. The prepared slides used in this lab are whitefish blastula preparations. A blastula is a mass of developing cells. After fertilization, the single cell continues through a series of mitotic divisions as it proceeds to maturity. One of the stages that it passes through is the blastula stage. While the whitefish is in the blastula stage, it is undergoing rapid cell division. Read the introduction to the lab before beginning any of the exercises. Handle prepared slides with care. Remember to use all lab drawing rules. Exercise 1. Mitosis in animals, whitefish blastula. 1. Obtain a prepared slide of the whitefish blastula. 2. The slides are a cross-section through the blastula. The cells will be round in shape rather than square in plants. 3. Locate the group of cells with the microscope on low power, then switch to high. 4. On high power, locate a cell that is not dividing. Its nucleus is in interphase. The nuclear membrane is clearly present and the chromosomes are not evident. 5. Draw what you see in the field of view. Title the drawing as interphase. Label the nucleus, cytoplasm, and the DNA. 6. Find cells that show the following mitotic phases. Title each drawing and label the chromosomes, or chromatids if the chromosomes have split. Label the cytoplasm and cell membrane. You won't be able to see it, but you know where it is. Now, find cells that show the following mitotic phases. Draw one of each and label the stage and what is occurring. Prophase. The DNA has duplicated and the chromosomes are starting to become visible. The nuclear envelope is disappearing or is entirely absent. Metaphase. The duplicated chromosomes are aligned in the center of the cell. Anaphase. The chromatids separate at the centromere, are beginning to move to the edges of the cell. Telophase. The chromatids are at the edges of the cell, and the cytoplasm begins to close in and separate the parent cell into two daughter cells. Exercise 2. Quantity of cells in mitosis. In this exercise, data will be gathered to determine the number of cells in the blastula that are dividing at any one time. 1. On high power, count all the blastula cells you see. 2. Now count the number of cells that appear to be in some phase of mitosis. Record your results on the table in handout. 3. Calculate the percentage of cells dividing by dividing the number of cells that are dividing by the total number of cells. Your answer should be a decimal less than 1.